Okay, I'm going to read this out loud so that you can follow along and take notes as I go in your student activity sheet, which can be found in the Nearpod slide. So, this is lesson 6, the inheritance of genes reading. And so how did the boys in the video get the mutation that resulted in DMD? So we're talking about the inheritance of genes. Part 1. Genes and alleles. What is a gene? Genes are segments of DNA that code for the production of specific proteins. The code of DNA is made up of 4 nucleotides that are named for the unique base making them up: adenine, thymine, guanine, and cytosine, ATGC. These 4 specific building blocks make up the letters of the DNA alphabet. The order of the DNA nucleotides is important for the production of a functional protein. So there are these 4 letters, and those letters make up the code of the DNA, and specific patterns of those letters give you the exact what the DNA says. How many genes do humans have? A genome is all of the genetic material in an individual cell of an organism. All cells in that organism contain the same genome. It is estimated that the human genome contains about 20,000 genes. Out of these 20,000 genes, over 100,000 different proteins can be made. This is because the coding regions in genes can be recombined with different segments to code for different proteins. There are over 6 billion nucleotides in the human genome, yet only 1.5% of those nucleotides actually code for proteins. The remaining 98% of nucleotides in the genome doesn't code for proteins. Some of the sequences in this portion of the genome work to turn genes on or off. That is, they control whether certain proteins are made or not. Okay, so to highlight, that's every cell has that whole DNA system that you have, right? And there's like 20,000 genes or sections of the DNA that make you who you are, that build the proteins that you need. And out of those, there's 100,000 proteins that are made. Okay? So just you can reread this and think it over. So what about alleles? What are alleles? Well, each cell actually contains 2 copies of each gene. The copies could be identical, or they could be different versions of the same gene, called alleles. For example, everyone has a pair of genes that code for blood type. A person with type AB blood has one allele that codes for A-type blood cell proteins, and another allele that codes for B-type blood cell proteins, while a person with type O blood has 2 alleles that don't code for proteins at all, so the proteins are missing. Okay, part 2, chromosomes. So what is a chromosome? Chromosomes are structures located inside the nucleus of animal and plant cells. Each chromosome is made of protein and a single molecule of deoxyribonucleic acid, DNA, which is condensed and folded around a protein. What do chromosomes do? The unique structure of chromosomes keep DNA tightly wrapped around spool-like proteins called histones. Without such packaging, DNA molecules would be too long to fit inside cells. For example, if all the DNA molecules in a single human cell were unwound from their histones and placed end to end, they would stretch 6 feet. For an organism to grow and function properly, cells must constantly divide to produce new cells to replace old, worn-out cells. During cell division, it is essential that DNA remains intact and evenly distributed among cells. Chromosomes are a key part of the process that ensures DNA is accurately copied and distributed in the vast majority of cell divisions. Still, mistakes do occur on rare occasions. Changes in the number or structure of chromosomes in the new cells may lead to serious problems. For example, in humans, one type of leukemia and some other cancers are caused by defective chromosomes made up of joined pieces of broken chromosomes. It is also important that reproductive cells, such as eggs and sperm, contain the right number of chromosomes and that those chromosomes have the correct structure. If not, the resulting offspring may fail to develop properly. For example, people with Down syndrome have 3 copies of chromosome 21 instead of the 2 copies found in other people. Do all living things have the same types of chromosomes? Chromosomes vary in number and shape among living things. Most bacteria have 1 or 2 circular chromosomes. Humans, along with other animals and plants, have linear chromosomes that are arranged in pairs within the nucleus of the cell. The only human cells that do not contain pairs of chromosomes are reproductive cells or gametes, which carry just 1 copy of each chromosome. When 2 reproductive cells unite, fertilization, they become a single cell that contains 2 copies of each chromosome, or zygote. This cell then divides, and its successors divide numerous times, eventually producing a mature individual with a full set of paired chromosomes in virtually all of its cells. How many chromosomes do humans have? Humans have 23 pairs of chromosomes for a total of 46 chromosomes. In fact, each species of plants and animals has a set number of chromosomes. A fruit fly, for example, has 4 pairs of chromosomes, while a rice plant has 12 pairs of chromosomes, and a dog 39 pairs. So here's a visual of the fact that different creatures have different numbers of chromosomes. Those are just little bundles of DNA, right? And each one has a different amount of them. So whether humans, you can see we have 46, and chimpanzees have 48. Dogs have 78. So we all have different numbers of those little chromosome bundles. Okay, so chromosomes are inherited. In humans, most other complex organisms, 1 copy of each chromosome is inherited from the female parent and 1 from the male parent. That explains why children inherit some of their traits from their moms and other from their dads. Do males have different chromosomes than females? Yes, they differ in a pair of chromosomes known as the sex chromosomes. Females have 2 X chromosomes in their cells, while males have 1 X and 1 Y chromosome. Inheriting too many or not enough copies of sex chromosomes can lead to serious problems. For example, females who have extra copies of the X chromosome are usually taller than average and have some mental retardation. Males with more than 1 X chromosome have Klinefelter syndrome, which is a condition characterized by tall stature and often impaired fertility. Another syndrome caused by imbalance in the number of sex chromosomes is Turner syndrome. Women with Turner have 1 X chromosome only. They are very short and they usually do not undergo puberty, and some may have kidney or heart problems. Okay, so now we're at part 3, meiosis. What is meiosis? Meiosis is a process that happens in certain cells that will divide to produce sex cells, also known as gametes. The cells that will go through meiosis are located in the ovaries for women and in the testes for men. Recall that there are 23 pairs of chromosomes in all human body cells, also known as somatic cells, making a total of 46 chromosomes in each cell. The goal of meiosis is to separate each pair of chromosomes in 2 new cells, each cell having 1 chromosome instead of 2. How does meiosis begin? The first thing that takes place prior to meiosis of the special ovary or testes cells is DNA replication. Each DNA molecule making up a chromosome is split between the base pairs, forming 2 parent DNA strands. Free nucleotide with complementary bases are matched to each parent strand. By the end of DNA replication, every chromosome has made its own copy, which is then attached to itself. Anytime DNA is replicated, there is a chance that the mutations can occur. The wrong base could substitute for the correct base, or a base could be deleted or inserted. After replication, the DNA molecule gets unpacked very tightly into a condensed chromosome with an X-like shape. Basically, what's happening in meiosis is that the sex cells only have one chromosome instead of a pair of two. So they're like a half cell, and they're just getting ready to come together and switch. So how are the chromosomes pairs separated in order to create two new cells? An amazing thing that happens once the DNA is packaged into chromosomes, each chromosome matches up with its partner chromosome carrying the same genes. Each pair of chromosomes then embraces, and segments of one chromosome switch places with the similar segments of its partner. This exchange of genetic material is called crossing over. Crossing over creates chromosomes with a different combination of alleles than originally inherited from one's parents. After crossing over, the paired chromosomes, homologous chromosomes, all line up in the middle of the cell. The random lining up of homologous pairs at the cell's middle is called independent assortment. From there, each chromosome of the pair is pulled by special fibers to the opposite sides of the cell. The cell then divides into two. At this point, the two cells that have been created, called daughter cells, each contain just one of each chromosome, one set instead of two. However, each chromosome is still duplicated because DNA replication happened at the beginning. The next part of meiosis is to separate the chromosome copies. In each daughter cell, the X-shaped chromosomes line up in the middle of the cell. The two sides of the X's are then pulled by fibers to the opposite sides of the cell. Each daughter cell divides once more so that the end results of meiosis is 4 daughter cells, each with one set of single chromosomes. We call the daughter cells with just one set of chromosomes gametes. What happens to the gametes after meiosis? Sperm and egg are both gametes with one set of chromosomes each. When the two join, their chromosome sets combine, and the chromosome number is now the same as the original cell that started meiosis in the first place. The new cell that is formed is called a zygote. It will divide again and again until it becomes a new offspring. When the zygote divides, does it do meiosis or something different? Almost all of the cells that descend from the zygote are called somatic cells, from the Greek word soma, meaning body. Somatic cells are cells that make up all of the body except for the gametes. Gametes are known as germline cells. Germline cells transfer genetic information from one generation to the next. Both somatic cells and germline cells replicate their DNA prior to cell division. As you read earlier, the cells that create gametes go through meiosis, a special type of cell division that reduces the chromosome number in half. We wouldn't want that to happen to all body cells, so somatic cells go through a cell division process called mitosis. How does mitosis work? Mitosis is like meiosis but simpler. The homologous chromosomes don't pair up. There is no crossing over, and there aren't 2 rounds of cell division. In mitosis, each chromosome replicates and then condenses into the compact X-shaped form. The chromosomes line up in the middle of each cell. Each copy of the chromosome is then separated from the other and pulled to opposite sides of the cell. After this, the parent cell splits in the middle to form 2 daughter cells, each with the same number of chromosomes as the parent cell had at the start of meiosis. Okay, that's it. You can go back and listen to it again, or read it on your own, the parts that you didn't understand, or ask me questions.